Certainly, Robert. Thank you for an excellent introduction. The first thing I'd like to do is to check with the participants today. How many of you already have any experience with NEURON? Any at all, ranging from nothing to a lot. Raise your hand if you can, please. Okay. Oh, I see more hands coming up. Excellent. Let's see, who's on the second page here? Wow. Okay. Well, that's very good. How many of you would say you just have very little experience or none? All the experts can drop their hands at this point. Now, Robert, your hand is still up. Okay. All right. Well, we're going to try to do something about that today. We'll try to help you with what you already learned, what you already know, and try to extend that, and try to expand your experience and knowledge if you don't know very much at all. Okay, so the first thing we're going to do is I'm going to share my screen. And this is our first slide. And what is it that we mean by neural modeling, and why are we interested in it? Well, basically, NEURON's core domain of applicability is in models in which the computations are being done by the generation, propagation, and interaction of electrical and chemical signals that are distributed in space and time in a biological structure, like the brain, the spinal cord, peripheral nerve, even muscle. A situation in which one needs to use computational modeling to test hypotheses about how function emerges from the properties of the cells and tissue, because the equations that describe these processes are just too difficult to have clear intuitions about. So that's the domain, that's a center of NEURON's domain of applicability. And the topics we're going to cover today include some introduction as to how to create models of cells and networks of neurons, a bit about how NEURON works, because in science, and it often turns out that it's best, you get the most out of a tool if you know something about how it does what it does. And we're going to talk about how to expand NEURON's capabilities by adding things that it doesn't come with when you get it out of the box. So new ion channels, new synaptic mechanisms, new types of chemical signaling and interactions, and artificial spiking neurons, etc. All right, so let's step back from the immediacy of all the technicalities and think about how we gain understanding about physical systems. In real world, physical systems are enormously complex and can be analyzed at many different levels of abstraction. And we typically have to, before we can begin to understand a physical system, we have to reduce this complexity. We have to find a way to make simplifying assumptions that are more easily grasped than the entirety of all the different properties of the physical system, ranging from submicroscopic at the atomic level or level, subatomic level, all the way to levels of behavior. So we make abstractions and simplifications when we think about biological systems, okay? We leave a lot of stuff out. And that's how we formulate conceptual models, okay? And we do this in physics and engineering as well. You leave out details, okay? So that you can have a concept that is compact, that is reasonably complete in a particular context that you can talk about. And if that conceptual model is straightforward and simple enough to analyze, you don't need to get into a great deal of mathematical rigor with it. You can just have clear intuition. So for example, if you're crossing the street and a large truck is barreling down the road at you, you don't need to be a trauma surgeon or a particle physicist to know what's going to happen if it hits you, okay? Our daily experience gives us very clear intuitions about that sort of thing. If, on the other hand, you've got a system that is very complicated and you don't really understand it, you understand the little individual parts of which it's made, but you may not understand all of them in great detail, and you may not understand how they interact because of their nonlinearities and the fact that they're distributed in space and time, and they're all interacting with each other, which is the case for most evolved biological systems. Then you need some intuition help. That's the point at which you need a computational model, okay? And what you're doing here is you're mapping your conceptual model into a computational form. You're representing your concepts by equations, and you're somehow getting those equations into a computer so the computer can solve them for you and produce results that allow you to explore what's going on in the model. Okay, and that's the role of computational modeling. And that's what we're going to talk about today. So on the left, I have a side view of a pyramidal cell from the CA1 region of hippocampus. And this thing has enormous anatomical complexity and also has tremendous biophysical complexity as well. There's channels spread all over the surface of this thing. There's synapses all over the place. The cytoplasm has all kinds of buffers and calcium storage and release mechanisms, active transport in the membrane, all kinds of stuff going on. But if what we're interested in is electrical signaling, we can start to leave out a great deal of that complexity. And if in particular we're dealing with a particular kind of experiment, let's say we're just poking the cell body and we're injecting current into the cell body and we're just seeing what happens to the membrane potential in the cell body as a function of time, this enormous complexity can be reduced to something which is anatomically quite straightforward and simple, this ball and stick model. And ball and stick models have been fit to biologically complex cells for decades now. I'm not sure it's much of a useful exercise anymore, but people always talk about the input impedance of a cell and something about its input resistance and how it charges when you inject a current pulse. So to some extent, people are still doing this stuff. And this ball and stick, even though it's a simple model anatomically, and it's described by simple equations, the solutions to those equations aren't that simple. They involve hyperbolic functions. There was a time when any engineer, mathematician, or physicist could on a sheet of paper sketch for you all the different hyperbolic functions and had very clear intuitions about the nature of the solutions to the equations that describe this ball and stick model. But that time is gone. We all rely on computers and calculators now to do graphs for us. So our intuitions aren't so clear even about this thing. So how do we get a computer to solve the equations that describe even this very simple conceptual model? Well, in Python, using NEURON, we can create a neuron model in which we represent the soma and the dendrite, and we've connected one end of the dendrite to an end of the soma, and that gives us a model that then the computer can solve the response to various input stimuli. The equivalent Hoke code is down here, and I include the Hoke code for reference for those people who may be going to labs where there is legacy code in Hoke, and they have to be able at least to read it, okay? But you can see from either of these little blocks of code, this is very compact, very human readable. You know immediately you get some inkling about what's going on by executing these statements, okay? All right, so that's an awful lot of simplification we went through here, and we've now represented these things in the computer, and it turns out that this representation can be extremely close match to the conceptual model. And why is that important? Well, let's go back to our original figure here. If we're going to use our computational model to get an idea of what our conceptual model implies, to evaluate the validity of our conceptual model, there needs to be a very close match between the computational model and the conceptual model. We did all our simplifications in this first step, forming our hypothesis about what's important in the biology to account for the phenomena we're studying. When we have something that taxes our intuition and we need to use a computer, what we put in the computer has to have a close match to what's in our head, or what's in the computer is not going to be useful for evaluating the implications of our conceptual model. And so Neuron makes this very easy. In fact, that is the strength of all domain-specific modeling environments, is that they allow you to make a close match between what's in the computer, okay? Unlike, say, a high-level programming language like C or naked Python, where you yourself have to create all the representations at a very low level. Okay, let's move ahead. What kind of models can Neuron handle? Well, obviously, it can handle a single compartment or a stylized, like ball and stick model, no matter how elaborate the sticks are. It can also obviously handle large-scale anatomically detailed models with many, many branches. There are many publications that report work that use such models. It can handle complicated network models. It can also handle models that have a great degree of mechanistic complexity. That is to say, have a wide range of different kinds of voltage and ligand-gated currents that include all kinds of synaptic transmission modalities, that may include gap junctions, that may involve extracellular fields. Neuron can also, you can use to add linear circuits to your models if you, say, are interested in instrumentation artifact, for example, and you want to see what happens if your electrode resistance varies or your electrode capacitance isn't properly compensated. How does that affect your recordings? Neuron can also be used to simulate the effects of diffusion, buffers, transport and exchange of solutes and second messengers. And it can also include models in which there is artificial spiking cells. Some people call these integrate and fire type cells, but our artificial spiking cells can also be included in a Neuron model. All right, so much for that. Let's now think about what it is that we're representing in our models. We're representing the generation and propagation and interaction of electrical and chemical signals. Well, now with electrical signals, what is it that's moving in the cell? Charge carriers, ions. What drives these, what drives purely electrical signals is electrical potential gradient. And what is conserved is charge, okay? Likewise, in chemical signaling, what's moving is solutes of various kinds. And what drives the movement of solutes is chemical potential gradients, or crudely concentration gradients, okay? And what is conserved is mass. Well, it turns out the equations that describe the spread of electrical signals are of exactly the same form as the equations that describe the spread of chemical signals. And so going forward, we're going to focus just on how Neuron deals with electrical signals, realizing that at the level of mathematics, chemical signals need to be represented in exactly the same way, and Neuron does that, okay? So let's consider then the basic equation that Neuron deals with, which is the conservation of charge. Let's say we have a small region of a cell. so compact that the membrane current density can be regarded as uniform over the entire surface of this little region. Okay? And we see these arrows represent transmembrane current leaving this region. If this, let's say this region has got neighboring areas that can contribute axial currents that flow into it. These arrows represent the direction of positive charge movement for axial currents. The arrowheads represent the direction of movement for positive transmembrane currents. And the equation, the charge balance equation for this little part of a cell is right here on the right-hand side. We have the sum of membrane capacitive current and transmembrane ionic currents must equal the axial currents that are entering the region, okay, from neighboring parts of the cell. All right? Very simple looking first order differential equation. Now what happens if you've got a cell with significant electrical extent? So there isn't just this, there's a whole bunch of regions like this. Well, to mathematicians, this would be a perfectly fine way to represent it. You just have a bunch of different compartments that have uniform current within each compartment, okay? Each compartment has its own membrane potential, its own membrane capacitance, its own transmembrane currents, and each compartment's got its own neighbors. So the jth compartment has got neighbors K, and the axial currents that enter compartment J from neighbors K is the sum of all these individual currents, where the resistance is the resistance between compartment J and one of its neighbors. And VK is the membrane potential of that particular neighbor, okay? And this is fine, except that biologists will look at this and say, well, wait a second, I'm really not interested in indices and numbers. I'm interested in parts of a cell that I can see under a microscope and think about. I want to know about dendrites. I want to know about spines. I want to know about somas and axons and axonal branches and presynaptic terminals. Give me something that looks like that. Well, that's what NEURON does. It allows us to think in biologically familiar terms. So imagine this bizarre looking cell. This is a real cell, an interneuron from hippocampus. And what we do is we take this and we break it apart at each of its branch points. And each of those pieces that we get by breaking it apart is like, it's like a cable. It's roughly cylindrical, at least on the small scale. And it is connected to a parent, and it's got child things that attach to it. And so each of these things is what we would call a section in NEURON, okay? All right? And so a section is NEURON's way of representing a continuous length of unbranched cable. All right? And if we have branches that are so long that we have to break them into pieces because transmembrane current density is not the same along the entire length of it, well then, we can do that. So the mathematical description of a section is basically one of these little compartments, many of these little compartments connected end to end, okay? And what a new section gives us is the cable equation for that particular compartment. And we don't have in our section anything else. We have just the membrane capacitance and axial resistance. There's no ionic current represented here. How do we add ion channels and pumps and reactions? Well, we do that. We have to just, we do that in different ways depending on what is the nature of the thing we're dealing with. If we're talking about ion channels that are distributed over the surface of the cell, we call those density mechanisms. Ion accumulation mechanisms are also density mechanisms because when ions enter the cytoplasm, well, it's kind of in the entire length of the section eventually. It's going to get distributed throughout the section. Synapses and electrodes act like point sources of currents, and we call those point processes. Don't get that mixed up with what a physicist or mathematician might call a point process. And these things can be described by differential equations or kinetic schemes or algebraic equations if we don't have to we be concerned with time. And we can build them with N-model. We can create new instances of ion channels or ion accumulation or other things like electrodes and synapses by writing N-model code. Or we can add new channels, new voltage and ligand gated channels with the channel builder. Or we can use the RxD module, which Robert is going to talk about later today. Okay. All right, we've talked about how we add things to sections. We've talked about the fact that a section is basically a cable and an unbranched cable. And so how do we use this? How do we use these concepts when we write code? Well, here are two examples of code that create a ball and stick model. The one on the left is the Hoke statements, and the one on the right are the Python statements. And the model that you would create would be exactly the same. It would have a soma, which has a length of 50 it's like a length of 50 microns and a diameter of 50 microns. Think of that, that's a cylindrical cable. The surface area of that is exactly the same as the surface area of a sphere with a diameter of 50 microns. And the soma has the Hodgkin-Huxley mechanism. And since the soma is electrically compact, we only need one compartment to represent electrical signaling in that soma. Okay. The dendrite is 200 microns long, got a diameter of 2 microns and a length, and we chop it into 3 pieces along its length because it's too long for it to be properly handled by a single compartment. Okay, the transmembrane current density is going to vary along the length of this. It has passive membrane properties, and the reversal potential for that passive linear mechanism is going to be minus 65 millivolts. And we'll notice that one end of the dendrite is attached to the soma. In fact, it's the zero end of the dendrite. Okay, what do I mean zero end? Let's go back to our picture of our cell. Each of these branches has an end that's proximal to the soma and an end that's distal to the soma. Why the soma? Because the soma is usually, at least in vertebrate animals, the soma is sort of the privileged section. It's near the spike trigger zone. It's usually the biggest part of the cell. It's the part you can hit with an electrode easier than any other part. And it's where usually the decision is made as to whether a spike is going to occur. Well, if not in the soma, then at least in the initial segment of the axon that arises from it. Okay, and each of these cables has got 2 different ends. Well, we treat the zero end as the more proximal end, and we call the distal end the 1 end. Okay, so each of these has a 0 and a 1 end. Proximal is what we call 0, and distal we call 1. Yes, you can be perverse and make models in which the proximal end is labeled 1, but we advise you not to do that. You're going to be causing problems for yourself. Okay. All right. So we now get into the notion of variables that can vary in space. What are these? An example is diameter. All these sections, neurites don't have the same diameter along their entire length necessarily. They can vary. So our sections can have diameters that vary along their length. Specific membrane capacitance can be a function of position along a section. Specific conductance of any particular ionic species can be a function of position along the length of a section. And of course, membrane potential can be a function of position. These are just some of the examples of range variables. There's many others. Transmembrane currents, for example, are typically range variables. All right? Okay, so how do we talk about range variables in a section? Well, we talk about it in terms of normalized distance along the length of a section. Okay, and this concept range has a value that can range from 0 to 1. It's 0 at the 0 end, and it's 1 at the 1 end. And in between, it has a value that is proportional to the distance from the 0 end. Okay, in fact, it's the relative anatomical distance from the 0 end. So here, if we had a neurite that's, say, 200 microns long, and we had something that was located at 150 microns distance from the 0 end, the normalized distance would be 0.75. That would be the range of that point, okay? All right. Now, since we're dealing with things that are distributed in space and we need to be able to map this into a digital computer, we have to deal with the fact that you can't represent a continuous, anatomically continuous structure in infinitely fine detail. So even though conceptually we think about range as being a continuous variable, we ultimately have to decide at what points in space are we going to be calculating the values of membrane potential as a function of position, or of any other range variable as a function of position. So this is where spatial discretization comes in. We can't really solve the cable equation numerically. It's a partial differential equation. We can't really solve it numerically directly because especially if it's got voltage gated channels, because of the complexities of the equations that result. Instead, we have to chop it up into pieces and represent each of those pieces, what's going on in each of those pieces, with a set of ordinary differential equations. That's the purpose of spatial discretization. We're making an approximate representation of continuous space, and the quality of that representation depends upon how fine we chop space up. So, if we have a section which is electrically compact, we can represent the membrane potential and the transmembrane currents and the parameters of that section with a single compartment. We set the section's nseg parameter to 1, and that means we'll be solving ordinary differential equations located at one point in space, and we'll be calculating the potentials at the 0 and 1 ends by algebraic equations, which are essentially cost-free. They impose very little overhead. The differential equations are in the middle of our segments, okay? If we need a finer representation in space, we'll be chopping our sections into more pieces. You'll notice that, and as we do that, that may change the locations at which we're calculating the values of membrane potential and segment currents. So if we change nseg to 2, you'll notice that we'll still be calculating membrane potential at the 0 and 1 ends, but now instead of calculating membrane potential in the middle of the cable, halfway down the length of it, and transmembrane current at the middle of the cable, we'll be calculating it at these two different points. Okay? And that's a problem. If we are trying to decide how fine do we have to chop space, because that's an empirical question. The answer depends on your model. Basically what you do is you run some simulations, then you chop space more into finer pieces, and you see if it causes a qualitative change in simulation results. And if it doesn't, then you know your original spatial grid was adequate, okay? Your original discretization was adequate. If it does, you know you need to try again and see if you need even a finer spatial grid, okay? The problem with going from an nseg of 1 to an nseg of 2, you'll notice that the points at which we're calculating the solutions to the ordinary differential equations are now different. we're calculating here and here. Which of these 2 points do we compare against the solution here to see if there's a qualitative difference? Well, obviously it's nonsense. You can't compare either of them because they're being calculated at different locations in space. So instead we need to increase nseg by an odd number because that simply adds new internal nodes, internal locations at which we're calculating membrane potential and solving differential equations. To do that, it adds new internal nodes on the sides of existing internal nodes when you have lower values of nseg, okay? All right, so why are these points in the middle of segments? Because neuron uses the center approximation to the second spatial derivative, okay? And that approximation is second order correct at the location of the middle of a compartment, not at the either end, not somewhere in between, but at the middle of a compartment, okay? So we're using the central difference approximation to the second order term in the Cable equation, the second derivative with regard to space. Okay, so how do we set that for a particular section? Say we had a section called axon, then axon has a property called nseg, and the syntax would be axon.nseg equals, and then we could put the value that we want on the right-hand side of the assignment statement. That takes care of it. So if we wanted to test spatial resolution in a model, and we're using Python here to do this, we would iterate over all sections in the model. This statement iterates over all sections in the model. You'll see this when Robert talks about Python code and how to script neuron models in Python. And for each of those sections, we're multiplying the value of nseg by 3, okay? And then we'd repeat the simulation. So this is how we would evaluate, does our model have adequate spatial discretization or not? Do we need to chop space more finely? Okay? In Hoke, you can do the same thing with this statement. All right. Now for any range variable that we're dealing with, the syntax for referencing the range variable is going to be the section name with an argument which is the numerical value of range. That is, where along the section are you interested in it? And then a period and then range var. And you read this by saying in sec name at the location specified by range, I want to access the value of some variable range var. So an example, at the middle of dendrite, 0.5 is the midway location, is halfway between 0 and 1. I'm interested in membrane potential. So I want membrane potential at the middle of dendrite, okay? And remember, if we have an odd value for nseg, there's always going to be a node in the middle, okay? Halfway down the length, okay? The Hoke syntax would be dend.v parens 0.5, which is, that's what it is. And the shortcut in Python or Hoke is dend.v without an argument. And that will access membrane potential at the middle of dend, halfway down the length of dend. This statement iterates over all segments in dend. And for each of those segments, it prints the range, that is, where is the center of that segment? What is the actual anatomical distance from the 0 end of dend? Because remember, this is relative distance, that's normalized distance. This is anatomical distance. Dend has a parameter called L that says how long it is. And the product, of course, is then the anatomical distance from the 0 end. And this returns the value of membrane potential at dend in that location. Okay, because seg x tells you the range of the midpoint of the segment. So, okay, so for every segment, tell us the range value that corresponds to the midpoint of that segment. Tell us the anatomical distance of the midpoint of that segment from the 0 end, and tell us the membrane potential at that point. Now it'd be more, this is just strictly for didactic purposes, okay, to emphasize this sec name range var syntax, okay? But actually, if you were coding this yourself, this is the way you'd write it, okay? Seg.x times dendel, seg.v, because every segment has a membrane potential. And the value of the membrane potential in that segment is that segment dot v, okay? All right. Okay, well, here's a bunch of different variables that we need to know about before we go on. Whoops, okay. And their names in neuron and their units, okay? Notice that time is in milliseconds, distance is in microns, membrane potential is in millivolts, and its symbol is V. That current typically is called I, and its units depend on whether or not it's a density mechanism, that is like an ion channel that's spread along the length of a section, in which case it's milliamps per square centimeter, or a point process, which is like a synaptic connection, you know, like a synapse, which is in an anatomically compact structure usually, and that means it's electrically can be treated as if it's a point source of current, or say like an electrode, like a voltage clamp or current clamp, in which case the units would be in nanoamps. Okay, now why these particular units? Well, these units come, the milliamps per square centimeter come from classical electrophysiology. Membrane biophysicists use this as their typical unit. Nanoamps are obviously relevant to modern patch clamping. Okay, so it's a little bit odd that they might be such different orders of scale, but there's historical reasons why it is. Specific membrane capacitance is either specific or absolute. Specific if it's typical membrane capacitance. Absolute is if you had some particular very localized capacitance, say like an electrode that had capacitance. This would be the capacitance of an electrode would be in nanofarads. Okay, conductances are in units of Siemens per square centimeter if they're for distributed mechanisms, and microsiemens if it's a point process like a synapse. Okay. Cytoplasmic resistivity is always ohm centimeter. Resistance, like series resistance in a single electrode clamp, is always in megohms. Concentration is always in millimolar. Okay. Unless you have N-model code that uses some other concentration, in which case the N-model code has to have some kind of a scale factor built into it. Okay, well, we're done with that. I'm going to plow ahead if I don't see some questions. I don't see questions. All right. So